Morning, everyone. It's a pleasure to be here today and sharing the stage with a lot of people who I admire greatly and enjoy working with. I think I've won the fire alarm lottery, so I apologize for the interruption. So this morning I'm going to be talking about vaccine coverage and effectiveness in high-risk populations. I'll be talking a bit about some case studies that we've done in OpenSAFELY, but to look at this, but I thought I'd start by stepping back and starting with this question of what does it mean to be at high risk? And so this graph here will be familiar to many people here. It shows the relative hazard of mortality in the first wave of the pandemic. This is the major sort of figure from the Nature paper that's been alluded to a few times already. And what we're seeing here is a mixture of society and biology. The probability of exposure to infection, and then the probability of that exposure resulting in a severe outcome or mortality. So it's this confluence of these two factors here. And in terms of what we think of in terms of vaccination policy of high risk, we're generally talking about that second component of the clinical risk factors that shape the probability of progression to severe disease and mortality. So you can see here, within sort of 2 months of the first wave kicking off in the UK, we're seeing evidence of significant excess risk in kidney disease patients, people with organ transplants, and several other of these clinical risk groups. And so this sort of sets the scene as to the high risk being shaped by these clinical factors. And then in the background of this, we have the vaccinology equivalent of putting a man on the moon. 11 months delivering vaccines that are effective against disease. So here I've got some of the landmarks from the development of the Pfizer vaccine. So by end of April, we have phase 1 and 2 combined clinical trials with 60 healthy adults telling you that this new vaccination platform is immunogenic and safe. By the end of July, we have the phase 3 efficacy study, 43,000 people being recruited across multiple continents and giving us this first efficacy data showing that this is highly effective at preventing cases. But again, these are generally including healthy adults. In this large phase 3 study, we're beginning to see recruitment of people with underlying health conditions, but they're generally at the milder end in terms of that eligibility. And so by the 2nd of December, we have the MHRA approving the use of the Pfizer vaccine in the UK. So as I mentioned, a monumental achievement in vaccinology. And this is then underpinning the JCVI's recommendations for this phased rollout of the priority groups. And in general, it's quite a sort of simple age de-escalation with frontline health workers prioritized to kind of make sure that the people who are most exposed are protected and we can keep the NHS running throughout this period. But then in orange highlighted text, I've shown the two groups within this rollout that feature the clinical risk groups. So groups 4 and 6, where you're extending that age threshold to include these higher risk adults. And the conditions that are defined by the sorts of studies that I just showed you. These are the clinical conditions that put people at higher risk. So chronic respiratory diseases, heart diseases, kidney diseases, and also this immunosuppression group, which is, as Will just mentioned, has been an eligibility factor for persistent spring campaigns, as well as the autumn booster campaigns. But at this point that these priority groups are being defined, it's worth asking, what did we know about the efficacy and safety of vaccines in the high-risk groups that I've just mentioned? And the answer is not a great deal. I mean, so we know from the Pfizer trial, this box here has got some estimates from that phase 3 efficacy study that was out in December 2020. We know that in people with milder comorbidities, that we see this persistent efficacy. We also know that people with hypertension that we see about that 95% efficacy. But the more severe immunosuppressed groups that are being prioritized in this JCVI criteria, we don't know a great deal about. So that sets the scene of putting the emphasis on post-licensure studies of vaccine safety, uptake, effectiveness, and also disease burden. So how does the burden change as we move from a pre-vaccine era to a post-vaccine era? And so I think that's sort of my intro, sort of setting the scene for the sorts of questions we need to answer, whether in OpenSAFELY or with other platforms available. And in the context of these questions, you have evolving policy, so new campaigns mean new priority groups with new age thresholds. We have new variants emerging, and we also have altering health-seeking behavior over time. So Andrea's just highlighted low coverage from a recent campaign of about 50%. And this idea of vaccine fatigue is sort of sitting in the back of some of these issues. And our challenge then is, can we deliver research that supports improved vaccine coverage? Can we support policy decisions through our research? And also, I'm a biologist by training, so I want to understand more about vaccines and use electronic health records to improve our mechanistic understanding of vaccines. And so I'm going to give a couple of quick case studies of a few of these areas through OpenSAFELY. So after the vaccine was rolled out in OpenSAFELY, within weeks, the team was delivering these coverage estimates in these priority groups. And these were highlighting, so you've got the clinical risk groups coming into this graph. And so these were from the early days of the rollout, highlighting variation in coverage by ethnicity and deprivation status, but also in certain clinical risk factors such as severe mental illness. And these reports were delivered across in weekly reports from December 2020. So then the next question here is, that's what we're seeing in the population as a whole. But if we were to take a clinical risk group where there's been a lot of messaging around the importance of vaccination and the high risk of disease, do we see the same pattern? So we carried out a cohort study linked with the UK Renal Registry to define people with moderate to severe kidney disease, including dialysis recipients and also solid organ transplant recipients. And what we saw was that these patterns that we were seeing in the population as a whole are also seen in each of these subgroups of people with kidney disease. So a lower uptake in people of minority ethnicity or in a lower socioeconomic deprivation areas. And we also saw some of these effects such as severe mental illness being a risk factor for lower uptake. And in addition, we started to unpick the intersectional effects of ethnicity and deprivation. So this is a graph where the more I look at it, the more scandalized I feel, in that you see, in addition to this effect of minority ethnic groups receiving lower coverage, if you overlay the socioeconomic deprivation index on top of that, you see that that's having an additive effect with very low coverage in the minority ethnic groups in lower socioeconomic quintiles. So the additive effect of these two factors. And so, it's quite a mild. I was expecting something a bit more dramatic. I think I can just carry on. And so we shared these findings with the UK Kidney Association to help highlight some of the trends we were seeing from OpenSAFELY in this coverage. So we have also updated these to look retrospectively. So this is a coverage snapshot that we carried out in September 2023, looking back at who'd received vaccines in the last 12 months. And we can see this, that even in these groups that had been recommended for 2 booster doses a year, in the case of kidney transplant recipients and hematologic cancer patients, we're seeing gaps in that vaccine coverage up to 2023, and a clear effect of age on top of that. So this is a sort of coverage insights that we can deliver using OpenSAFELY, a granular insight in individual clinical risk groups. Secondly, a question about biology and mechanistic insights. So here we were using that same cohort of kidney disease patients and dialysis and transplant recipients and saying, we saw from immunogenicity data coming out from the lab that there was a gap in terms of the antibody levels induced by the Pfizer RNA vaccine relative to the AstraZeneca vaccine. But if a booster dose of the RNA vaccine seemed to level the playing field and close that gap. So that's a nice example of a lab's finding. But the question then is, does this actually, what's played out against clinical endpoints? And so this is where you can ask that question. Instead of having 60 people with a lab assay, you can then do it in 1 million people using OpenSAFELY health records. So we repeated this study comparing 2-dose vaccine effectiveness for the AstraZeneca and Pfizer vaccine, and then after that initial vaccination series, and then after a subsequent RNA vaccine booster. And we saw the same effect that we'd seen in that closing of the gap, where you saw this differential effectiveness after that primary series, but an RNA booster dose closed the gap. So this was a nice example of this mechanistic understanding being consolidated by a use of these electronic health records data. And again, we're delivering this, saying that this is across these granular subgroups, and not just people with moderate to severe kidney disease, but also in the dialysis recipients and also in the transplant patients. We're seeing that same trend. And so, yeah, so that's the validation of that immunogenicity signal using clinical endpoints. And finally, just trying to take this a step further and give contemporary data about the incremental effects of booster doses that have been given every spring and every autumn are being recommended in these high-risk groups. And so we've started to look at this in the groups I've shown here. So breaking down this broad umbrella of immunosuppression into these categories of organ transplant, compromised bone marrow, radio and chemotherapy recipients, and other subgroups, and reporting on the risk factors of severe disease in these groups. And we've presented these findings to JCVI a couple of months ago, and we can emulate this in the future. So finally, I just wanted to finish by looking forward and saying, all of these things that we're doing can continue to do. The vaccine program is expanding. Last year we had Shingrix rolled out to over 70s. This year we're seeing RSV vaccine implemented. RSV vaccine at over 75, so that creates a regression discontinuity. We can look at above and below the 75-year threshold. Shingrix was rolled out to high-risk immunosuppressed populations from 2021. We could do trial emulations to look at this. So everything we've talked about, these tools, can be applied to this expanding vaccine portfolio. So I think there's a lot of future scope for extending what we've learned and the tools we've built. And on that note, I think I'm overstaying my welcome. So I'll thank the many people who have helped deliver all this work. And thank you for your time.